going to have a quick revision of the chapter Plant Reproduction Part 1 of Class 12 Biology. I hope that you have a fair knowledge of the basics of it before we start over here. To begin with, we understand that reproduction is a very important process because it ensures the continuity of species generation after generation, and for flowering plants also it is a very important characteristic that reproduction occurs. So flowering plants show sexual mode of reproduction and they bear very complex structures as male and female reproductive units along with the accessory ones. So how do you define a flower? It's a modified stem which functions as a reproductive organ, and it's known for producing ova and pollen. Now as soon as we see the structure of a flower, the main things that we should notice are the stigma, style and the ovary for the female part, and the anther and the filament for the male part. These are the main parts that are involved in the sexual reproduction of any flower. So for the pre-fertilization events, there are several structural and hormonal changes, and they are seen to form and develop the floral primordium. And inflorescence is something that is bears floral buds and then flower. In flowers, both male and female parts are differentiated, and then male and female gametes are produced. Coming to the structural details, stamen, microsporangium and pollen grains. So stamen, as I already said, it has a long stalk that we saw as the filament, and another one as the bilobed anther. The top part that we saw, where it contains two theca. Each lobe contains two theca, that's why they are called dithecious, and that is known as the anther. So the anther is a four-sided structure, as you can see here. It's a four-sided structure and it consists of microsporangia, and it has two in each lobe. Microsporangia further develop and they become pollen sacs which contain the pollen grains, which play a fundamental role in the pollination process. So here, as we see, this is the anther and this is the filament, the stalk. As I described already, microsporangium has four layered walls: epidermis, the endothecium, the middle layer and the tapetum. So why is the tapetum important? Because it nourishes the developing pollen grains, so definitely it has a very important characteristic feature of the development process. So there are the sporozenous tissues which are compactly arranged and they are of the same type. So they are homogeneous cells, and they are found, where are they found? They are found in the center of each microsporangium when the anther is young. And the process of formation and differentiation of microspores, that is the pollen grains, from the microspore mother cells by a reductional division is called microsporogenesis, and this reductional division we already know is meiosis. So the cells of sporogenous tissues, they undergo meiotic division to form the microspore tetrad. So there are four different parts of it, so it's known as tetrad. And as the other anthers mature and dehydrate, the microspore dissociates and they develop into pollen grains. So as you can see in the figure, whatever I said is explained here: the middle layer, the connective epidermis tissues, the endothecium, sporogenous tissues. And if you magnify this part, a structure like this is visible under the microscope: epidermis, endothecium layers, these are the middle layers, and these are the microspore mother cells, the MMCs, which are further important. This is the tapetum, which is giving us the nourishment for the cells, and this is another part which gives us a diagrammatic representation of the pollen grains. So what are pollen grains? Pollen grains represents the male gametophyte. So as you know, it's a sexual reproduction process, so male gametophyte, female gametophyte have a very important important roles to play. So pollen grains have two layered walls. One is the exine, another is the intine. So exine, very interestingly, exine is an outer hard layer, and there is no substance, no known enzyme that is known to degrade the sporopollenin. Now this contains, the exine is actually made up of sporopollenin, and it can withstand high temperatures and strong acids and alkalis. So we can understand that if there is a lot of change in the pH also, it won't really matter here, because this is like absolutely not affected by presence of any kind of change in the external atmosphere. And therefore we can understand that since it is not affected, so how much protection it can actually give to the inside constituents. And the intine is the inner wall, which is a thin and continuous layer, and it is known to be made up of cellulose and pectin. So a plasma membrane surrounds the cytoplasm of the pollen grain. The mature pollen consists of two cells. So as you can see here, one is the vegetative cell, another is the generative cell. Very importantly, vegetative cell is the outer one, which is the bigger, and it has abundant food reserves, large irregular nucleus is there, and this is the generative cell, which is small and basically involved in the fertilization or the fusion process with the egg, and therefore it has dense cytoplasm and nucleus, because basic hereditary part has to be done by this generative cell only. Now pollen grains, we already know, example in Parthenium species, are known to cause severe allergies. So they are allergens and bronchial disease in some people, and which leads to the chronic respiratory disorders like asthma, bronchitis, etc. And the viability of the pollen grains, it actually differs from species to species. So it is very important to ensure that these pollen grains actually land on the stigma before they can germinate. So if we want the germination to happen, then it has to definitely land on the stigma. Pollen grains of a large number of species, they are stored in the liquid nitrogen at a temperature of minus 196 degrees Celsius. This is called the pollen bank, and the process of this storing is called as the cryopreservation. Let me also take a moment to introduce you to Missionet. It is an online tutoring and mentoring service which connects you to Tier 1 college students of IITs, NITs, AIMS, etc., from which you can ask any kind of doubts which you may have with regards to your competitive or board examinations. Their fees start from a minimal rupees 1000 per subject per month, where you get the said services along with AI-based feedback as well. You can reach out to them at contact@missionet.in for any queries. That marks the end of our session. Hope that was good. Make sure that you like the video and subscribe to our channel for further updates. Thank you.